Hello everyone, welcome to the ITLS Academy. Today we are going to discuss our topic that is your plant tissue culture. Okay, so before starting the lecture in this lecture, what should we are going to do? We are going to see the basic of this plant tissue culture. Okay, then after that we are going to discuss in the next lecture about the history of the plant tissue culture, and then the next level of the this program will be proceed. But in today's lecture, what should we are going to discuss? We are going to discuss the introduction part of this plant tissue culture. Okay, so basically, what is this plant tissue culture? Plant tissue culture is simply a technique in which, okay, it is simply a technique in which plant cell, plant cells, tissue, or organs can be grow in a controlled environment, can be grow in a controlled environment so that we get a desired property. Okay, in plant tissue culture, what happened? Simply, we grow either a plant cell, either a plant tissue, or either a whole plant. Okay, and it can be grow in a controlled environment so that the desired property will be achieved. Simply, it is a plant tissue culture. Okay, why it is useful? Because it can produce our desired property. Like suppose that it can produce a disease free plant, disease free plant. Okay, so this is our desire that we have to produce up some plant. Like suppose that there is a plant. Okay, now this A plant is what? There is some disease into it. Okay, now we have to produce this A plant as our this disease free. Now for this purpose, we have to follow the plant tissue culture. It also used to produce a high quality planting material. High quality planting material. Okay. That's why it is also useful. Okay, so there are different stages of the plant tissue culture. Okay, different medias are used in this plant tissue culture. So one by one, we are going to discuss all these things. But here in this lecture, we are only focus the basic of this plant tissue culture or introduction of this plant tissue culture. Okay, so let's start today's lecture. First of all, we are going to see the definition of the plant tissue culture. Okay, so simply that I already told you, the plant tissue culture is simply what? It is a technique which is used to produce a, okay, it is a technique which is used to produce a, which is used to produce a plant cell, tissue, or organ of our desired properties. Okay, either the plant are free from any type of disease, either these plant are what, they have a high quality of the planting material. Okay, and it can be grow uniformly and repeatedly. Okay, so simply it is a plant tissue culture. Now in plant tissue culture, different medias are used, different medias are used. Okay, so what should we do? We take a media and just a sample of the plant or a tissue of the plant will be used. Tissue of the plant will be used, and this hole will be performed in a isolated chamber. Means that the chamber which are free from any type of the contamination. Okay, so in that chamber we are going to produce this, and medias are used. This medias are what? These are the food for the tissue which we want to grow. Okay, then when it will grow into a media, then it will transfer into a field. Okay, so this thing that we are going to discuss. Now, see what is written in a slide. Plant tissue culture is the science of growing plant cells, tissue, or organs isolated from the mother plant. Okay, suppose that we have a mother plant. Now, what we have to do? We isolated from the mother plant on a artificial media in vitro under a control condition. Okay, so one thing is very important in a plant tissue culture. We provide a control condition. Okay, that's why we get our desired properties. Okay, now see here a tissue sample scrap from a parent plant. Okay, now this is your our parent plant. Now what should we do? We scrap a tissue with the help of the scrubber, and then this tissue will be transferred into a media. This tissue sample placed in a agar growth media containing a nutrient and auxins. Okay, nutrient and auxins both are for the growth of this plant tissue. Okay, now we transfer it, and then this sample are developed into a plantlets, and this plantlets are planted into a compost. Okay, and where we grow it. Compost is what? Compost is the area in which we control the environment. It is the it is the area in which we control the environment. Simply. Okay. So basically, this is about the plant tissue culture definition. Okay. Now come to the next slide. Now we are going to see the just a brief history of this plant tissue culture. Brief history. Brief history of the plant tissue culture. Now, first of all, everyone, everything just that know that everything have discovered by something. Okay, so we call Haberland a father of the plant tissue culture. Okay, and he conceived the concept of totipotency and cell culture in 1902. Okay, now first of all, who will call the father of the plant tissue culture? So Haberland, called a father of the plant tissue culture, he conceived the concept of totipotency and cell culture in 1902. Now again here a one word, that is your totipotency. What is this totipotency? Totipotency is the simply the ability of the cell that it can be divide. Ability of the cell that can be divide. This is called a totipotency. Ability of the cell, single cell, to divide and produce all of the differentiated cell in an organism. Now, this Haberland was the first who considered that the culturing cell aseptically in a nutrient solution. Aseptically means that the environment which are free from a microorganism. Environment which are free from any type of microorganism. Okay, so he's the first one who considered that we can culture a plant tissue in a aseptically inside a nutrient solution. However, he was unsuccessful in his culture as he failed to recognize that the cell differentiation required plant growth regulators. Okay, so along with the nutrient solution, we also required a plant growth regulator. Okay, for the differentiation of the cell. But Haberland cannot recognize that the for the differentiation of the cell we required a plant growth regulator. So at this point he is failed. But on the other thing that the father of the plant tissue culture is considered Haberland. He also gave a concept of this totipotency. He also considered that the for the culturing of the cells we required a aseptically nutrition solution. So basically for the culturing of the cell we require two things. One is your nutrient solution which are aseptically, and the another thing is plant growth regulator. Okay, so one thing was described by the Hebeldine, but the other thing, that is your plant growth regulator, he cannot be recognized. Okay, now the next one is your types of the growth: organized growth and unorganized growth. There are the two type of the growth. One is your organized growth and another one is your unorganized growth. Now, this organized growth can occur plant, cell, or tissue are transferred to a culture media and continue to grow with their structure preserved. Okay, so this organized growth, what they do, they occur in plant cell or a tissue, and they will transfer into a culture media, and inside the culture media it is continue to grow and with their structure preserved. Now, the unorganized growth, it can occur in a pieces of whole plant which are cultured in vitro, and the cells are aggregated and they have a typically lack in recognized structure. Okay, in organized growth we recognize the structure because the structure are preserved, but in unorganized growth the structure are not recognizable. Okay, because their structure is not preserved. For example, callus and cell suspension culture. Callus and cell suspension culture are the examples of this unorganized growth. Now the next is your, see, this is your unorganized growth. You can see that here the structure are very clear. This is your leaf, this is your shoot, this is your leaves, this is your shoot. Okay, and here this is your root, and this is your branches. You can see that they are what? They are the organized. Every structures are preserved. Structures are preserved. You can recognize the structure. Now in the next, that is your the calcus. Okay, here you can see that you can recognize the structure are preserved. It can be grow by either part of the plant, but their structure cannot be organized. Their structure cannot be preserved. Structure cannot be preserved. Okay, you can see that here. Now the next is your ungrowth, sorry, unorganized growth of the suspension. You can see that here it is the suspension solution. Okay. Now in the suspension solution, you can also see that the growth cannot be recognized. Growth cannot be recognized. So these are the examples of the unorganized growth or organized growth, or you can say that the diagram of the Diagram of the organized and unorganized growth. Okay, clear? Now come to the next slide. Establishment of the tissue culture system. Okay, we are not perform this tissue culture system in anywhere. Like suppose that we have a lab and just go in a desk in a slab and just perform the experiment. It is not possible because when we performing this tissue culture system, we required a aseptically environment. Aseptically environment, which means that we required a environment which are free from which are free from any type of microorganism. So that type of environment we require. So for this purpose we have to establish a tissue culture system. Now the first one is your we have to select a X plant. Okay, so it can either a root, it can either a root, stem, leaves or bud, any part of the plant. You can select it. Either part of the plant, either it is root, either it is shoot, either it is stem, leaves or bud. Okay, you can select it. After the selection of the this part of the plant, you have to isolate it, and along with it you have to preserve it, sorry, sterilize it. What you have to do, you have to isolation and sterilization. We all know that means that the process in which a area or a thing free from any type of microorganism. Okay, now prevention of contamination of tissue culture media is very important for the whole process of plant. Okay, so for a whole plus process of this plant propagation, we have to consider one thing very important, that is your prevention of contamination. Okay, we have to prevent our contamination in all the steps, then this process will be a successful. So all the work should be performed in a special room or inside a hood or a cabinet from which microorganisms are excluded. Okay, so this whole process will be performed in a separate or a special room in which the microorganisms are excluded. Okay, because we have to prevent a contamination, that is very important. Okay, see here you can see that in a diagram there is a area in which it is what, it is a laminar, nothing special. You can perform it inside the laminar because the laminar are free from any type of contamination, but you have to take care. Okay, now the next is your suspension culture system. Okay, so first of all, explant selected and surface sterilization. Then callus culture induction on solid media supplemented with sucrose, hormones and agar. See, here I'm just telling you all the basic thing in detail, like how we suspend it, how we select the explant, how we sterilize the surface, how this calculus culture are inducts, and how the solid medias are produced. Okay, and what is the ingredients of the solid medias like sucrose, hormones, and agar? What is the function of all this? All these things will be covered in next classes. But here, what should we are going to do? We are just focus on a basic of thing. I'm just compiling everything. Okay, now Callus is introduced to our agitated liquid media. Okay, now what should we do? We introduce the scalate inside our agitated liquid media. Now the next is your hairy root culture. Okay, this type of culture are produced when a infection of the explant take place. Okay, and this infection of the explant are take place by a gram negative bacteria, that is your soil bacterium agrobacterium rhizogenus. Okay, this hairy root culture are what? They are a infection. Okay, and this infection are caused by a explant, and the bacteria which is responsible for that infection are soil bacterium agrobacterium rhizogenus. Okay, now this process are lead to the formation of the naturally occurring hair root disease. Okay, now when there is an infection caused, then there is a disease occur, that is your naturally occurring hair root disease. In a diagram here you see that a hair-like structure or form, hairy roots. This is what? This is the infection caused by a soil, gram-negative soil bacterium, agrobacterium rhizogenus. Now, it is the culture produced after the infection of explant and a culture by a gram-negative soil bacterium, agrobacterium rhizogenus. This process leads to the formation of naturally occurring hair root disease. Simply. Now, the next is your process. This is your agrobacterium cell. This is your plant cell. Okay, here here this is your integrated DNA. Here, what happened? This is your VR genes, diplasmid and tDNA. Okay, now this tDNA from a T stranded are transferred to the plant cell, and now here it is from a T complex. Now this T complex are transferred to the DNA and form a integrated T DNA. And basic all the things are same, like your wireless protein here. Okay, bacteria, chromosome, transported channel here. See, this is your what? This is your transported channel where the three strand are transport from a agrobacterium cell to a plant cell. Okay, here this is your mitochondria, chloroplast and nucleus. Now the next is your induction of this hairy root culture. Now explants are wounded and inoculated with a rhizogenes. After 2 to 3 days, explant transferred to a solid media with a antibiotic called cefotaxime or vancomycin. Okay. First of all, this explant are what? They are wounded and inoculated with a rhizogenes. Okay. Now, after 2 to 3 days, this explant are transferred to a solid media. And in a solid media, there is an antibiotic are present, which is called a cefotaxime or vancomycin. Hairy root will be induced within a 1 to 4 week, and it totally depend upon the species of the plant. But at least 1 to 4 weeks will be required for the development of this hairy roots. Now hairy roots are separated and cultured on a solid media until enough biomass is obtained. Okay, when we get enough biomass, then we this hairy root are cultured. Okay, now the next is your advantages of hairy root culture. The hairy root system is a genetically and biosynthetically stable. Okay, this hairy root are what? They are genetically and biosynthetically, in both way they are what? They are stable. They produce a secondary metabolites. Okay, and it can be grow under a phytohormone free condition. Okay, it can easily, it can be grow into a phytohormone free condition. This culture also show a fast growth, and it will reduces the culture time and easy to handle. Okay, so there are four advantages of this hairy root culture. One is it is genetically and biosynthetically stable, and next is your it produces a higher it produces a high higher production of the secondary metabolites, and it can also grow under a phytohormone free condition, and this culture show a fast growth which reduces the culture time and it can be easy to handle. Plant growth regulator. Okay, so this is very important for the um, culturing of plant tissue culture. For the culturing of this plant tissue culture, this plant growth regulator are very important because the nutrition are required, but along with the nutrition we also required a plant growth regulator. Growth and development of a plant culture usually depend upon addition of plant growth regulator to the medium. Okay, so here you see that here the also written because the nutrition is required, and this nutrition is get from a media, but along with nutrition this plant growth regulator will also required for the growth and development of the plant culture. They are important in plant tissue culture since they play a vital role in stem elongation, tropism, and apical dominance. They are generally classified into the following group, that is your auxins, cytokinin, gibberellin, and abscisic acid. This is a plant growth regulator or plant growth hormone. So basically we are using the auxin, but you can also use a cytokinin, gibberellin, and abscisic acid along with auxin. So these are the plant growth regulators. Now the next one is your moreover proportion of the auxins to a cytokinin. It determines the type and extent of the organogenesis in a plant cell culture. Okay, the proportion of this auxin and cytokinin, it will determine the type and extent of the organogenesis in a plant tissue culture. Suppose that if we have a higher auxin and less amount of cytokinin, then root will be developed. Okay, or in another language you can also said that for the development of the root we required a larger amount of auxin and this cytokinin in a less amount. Okay, or you can so say that when a auxin is increases and cytokinin is decreases, then this root will be developed. Okay, now the next is your when cytokinin is increases and auxin is decreases, then shoot will be developed. When the concentration of the auxin and the cytokinin are same, then the callus are developed. Then a callus are developed when the concentration of auxin and cytokinin are equal. Okay, now this is the first diagram. You see that it is a indole 3 acetic acid. This is your zeatin natural cytokinin. Now the next is your in vitro culture and production of important secondary metabolites. Now this secondary metabolites also play a very important role in the deficiency of this insect herbivorous, microbial pathogens and facilitating pollination and reproduction. Okay, now it totally depend upon the structure of this. The secondary metabolites are classified into a different group. Okay, either they are alkaloids, flavonoids, phenylpropanoids, terpenoids, steroids, tannin, and proteins. This this classification are based on a structure of the secondary metabolites. Okay, either they are what? They are alkaloids, flavonoid, phenylpropanoids, terpenoid, steroid, tannin, and protein. And it can play a important role in a elimination of the insect, herbivorous, microbial pathogens, insecticide, herbivorous, microbial pathogens, okay, and fasciculating the pollination and reproduction. Okay, it eliminates the insect, herbivorous, microbial pathogens, but it fasciculates the pollination and the reproduction. Okay, and these compounds are biosynthesized through a series enzyme which are catalyzed the just a minute. These compounds are biosynthesized through a series enzyme catalyze the reaction by using a simple building block in a different ways. Okay. Now the next is the main biosynthesis pathway, which included the first one is your shikimic acid pathway, that is your phenylpropanoids, mevalonic acid pathway, steroid and triterpenes, amino acid pathway, alkaloids, acetate pathway, fatty acid, and combined pathway, flavonoids. Okay, now the next is your strategies development to maximize the production of target compound. Now, the first strategy is expressing the key genes which involve in a biosynthetic pathway. Also block are competitive branches of the biosynthesizing target compound and increase the biomass of vegetarian growth and increase the production of the target compounds. Also introduce the key genes into a microbes and use a combi combinatorial biosynthesis, which is used to produce a target compound or important intermediates. Okay, so these are basically four strategies which is used to or which is developed to maximize the production of the target compounds. Now the next is your elicitor and signaling the pathway. Elicitors are the substances that can trigger the hypersensitive reaction in a treated plant cell. Okay, so simply this elicitor is what? It is a sample substances. It is what? It is a substances which can trigger a hypersensitive reaction in a treated plant. The plant which are treated, which are used in a plant tissue culture, it can trigger the hypersensitive reaction in that type of the plant due to the effective regulation of the gene expression and activation of the secondary metabolism. Elicitors are used widely in a medicinal plant cell and tissue culture to maximize the production of the target compound. Okay, so this elicitors are basically used in a medicinal plant cell or tissue culture in a medicinal view. Biotic elicitors, which include fungal polysaccharides, proteins, cell debris, and conidium. Abiotic elicitors, which include heavy metals, iron, UV light, osmotic stress, and even a sonication which been reported to have positive effect towards the improvement of the this secondary metabolites. Okay. Now the next is your precursor feeding. It is used to improve the yield of the secondary metabolites. Okay. For the improvement of the yield of the secondary metabolites in a plant tissue culture, we use a precursor feeding. And this precursor feeding is an effective approach. Now, the second point is that precursor are compound which are existed in a upstream of target compound in a biosynthetic pathway, and most intermediate can be used as a precursor. The concentration of a precursors determine the reaction speed. At higher concentration, the reaction speed is usually higher than when the precursor concentration is lower. Okay, if we use a higher concentration of this precursor, then the speed of the reaction will also increase. But if we use a lower concentration of this precursor, then usually the speed of this reaction will be decreases. Okay, now the next one is your bioactive secondary metabolites from a plant tissue culture. Okay, so here the plant name, then active ingredient in that plant, then the culture type, and here the references. Okay, so then the first plant is your Taxus species. Okay, first plant is a Taxus species. Active ingredients on a Taxus species is Taxol, and the culture type is suspension. Okay, and here it is, the references are there. Now the next plant name is your capsicum annum, and the active ingredients in capsicum annum in capsaicin, and the culture type is suspension. The third plant name is Canthanus rosaceus, and the active ingredient in that plant is indole alkaloids, and the culture type is suspension. Okay, now the next plant is Ephrida species, and the active ingredients is L-ephedrine, and the culture type is suspension. Okay, now the next plant name is Cassia acutifolia, and active ingredient in that plant is anthraquinones, and the culture type is suspension. Okay, now the last plant name is your Coffea arabica. Active ingredient is caffeine, and the culture type is callus. Okay, now the next is your production of taxol from our taxol species. This taxol is what? It is a complex diterpene anti-cancer alkaloid drug. Okay, taxol is what? Taxol is a complex diterpene anti-cancer alkaloid drugs which are found in 1971 by a Vanni et al. From a Pacific E tree, Taxus brevifolia. Okay, and at represent the drugs is approved for a clinical treatment of ovarian and breast cancer by the FDA. Now, FDA approved that drugs for the treatment of the ovarian and the breast cancer. However, the supply of the Taxol for a clinical use is only limited. Either it is natural source or by total synthesis. Optimization of the culture condition. Dark conditions are suitable for the growth of the cells and Taxol production. Okay, abiotic alcylator from a Rhizopus stolonifera fungus around 25 milligram per liter will be used in the combination with the abiotic acetol methyl jasmonate 10 milligram per liter and salicylic acid 100 milligram per liter are shown to improve Taxol production around 16-fold when it is added at a day of 25 to 30 of a culture growth medium. Okay, so this is the optimization culture optimization for the culture conditions. Okay, Now, next one. Supplementation of the medium sucrose, phenylalanine, and ammonium citrate, which are result in 5.6-fold higher taxol production. Okay, if we use a supplementation of the medium. Okay, so if we use this supplement of the medium like sucrose, phenylalanine, and the ammonium citrate, then the production of Taxol is around 5.6-fold higher, which is compared to a control like 13.75 milligram per liter as compared to the 2.5 milligram per liter if we are not using this supplement. Now, currently, bioreactor up to 75,000 liter are being employed for the commercial production of the paclitaxel from a cell culture by a Phyton Biotech, ESC Agenetic, Samyang Janis, and Netherman, Germany. Okay, now the next is your production of capsaicin. Okay, see, capsaicin is what they are alkaloids, and they are obtained from a capsicum species, and it is mainly used for the it is mainly used as a pungent food additive in a formulated food. Capsaicin is also used in a pharmaceutical preparation as a digestive stimulant for a rheumatic disorder. Okay. Now, this suspension culture of the capsicum frutescens, it produces a low level of capsaicin, but immobilizing the cell in a reticulated polyurethane foam, which can increase the production approximately 100-fold. Supplying the medium with 2,4-D, that is 2 milligram per liter, carnitine for 0.5 milligram per liter, and sucrose 3% are the optimum condition for the capsaicin production. It further improve can be brought about by supplying a precursor such as a isocapric acid. So this is all about the basic of the plant tissue. I hope you will understand. If you have any query related to this topic, you can ask me in a comment box, or for a further query you can follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Or for a further query, you can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450. Visit our website www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.